In this part of the course, we're going to be covering the various different kinds of organisms that we share this planet with. In this lecture, what I'm going to be talking about are the smallest of the organisms that are on this planet, the simplest, perhaps, depending on how you classify simplest, and certainly the most abundant. We're talking about the prokaryotes, the bacteria and the archaea, two groups that lack intracellular organelles or nuclei. When I say that they're abundant, that's rather understating the case. There are about 10 to the 30th power prokaryotic cells on this planet. That's not just a billion billion, but a billion 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 and then some. Not only do prokaryotic cells outnumber all of the eukaryotic cells on this planet, they probably outweigh them as well. This despite the fact that prokaryotic cells may be 1,000 times smaller than eukaryotic cells. You have a very close personal interest in these prokaryotic organisms as well. Depending on who you talk to, someone may tell you that there are actually more prokaryotic cells in your body than there are eukaryotic cells, that is, your own cells. Many of the bacteria that live in you and on you are incredibly important for your health and well-being. They produce vitamins, they keep pathogenic bacteria at bay, they help you digest things. Your mouth, right now, is home to something like 300 species of bacteria, with a few protozoa and fungi thrown in for good luck. So these omnipresent organisms basically can't help but be important not only to us, but to the ecology of the rest of the planet. One thing I want to emphasize is, although we've called them all bacteria for most of the history of science, it's recently been recognized that the prokaryotes on this planet are divided into two groups that are not even one another's closest relatives. The bacteria and the archaea are both prokaryotic groups, that is, they lack a nucleus, but this is a plesiomorphic character state. That is, it's an ancestral character state. The eukaryotes, of which we and all other animals, as well as plants and fungi, are members, have a true nucleus, but we are actually more closely related to the archaea than we are to the bacteria. There are some morphological differences between the bacteria and the archaea, but they're quite subtle. And the reason that until recently the archaea weren't recognized as being separate from the bacteria is that there weren't many morphological traits to go on. We only really appreciated that there were so many archaea out there and realized that they were more closely related to eukaryotes than to bacteria when we started being able to sequence their genomes. Nevertheless, you can find a few differences between bacteria and archaea. Bacteria all have a cell wall that looks something like this. This is an electron micrograph of the cell wall. It contains a carbohydrate called peptidoglycan. Peptidoglycan, like a lot of different structural carbohydrates, can be linked together in long chains, and those chains can then be cross-linked into a sheet. And that's what makes up the resistant cell wall of a bacterium. Archaea also have a cell wall, but it does not contain peptidoglycan. Eukaryotes, as you'll learn in the next lecture, don't have a cell wall at all. So this is not a synapomorphy that they share with the archaea. Another way that archaea and bacteria are different is they have different kinds of cell membranes. But in this case, the archaea have a cell membrane that is different from both the bacteria and the eukaryotes. If you went solely on this trait to try to determine common ancestry, you would conclude wrongly that the bacteria and the eukarya are one another's closest relatives. The archaean cell membrane, in this particular instance, is an autapomorphy. something novel, but something that isn't shared between any of the three groups, or between any two of the three groups that we're looking at. Another difference between bacteria and archaea, and one that does unite the archaea with the eukaryotes, is the presence of histone proteins. These may seem trivial. There's these little kind of green balls that the chromosomes get wound around when they're not actually in use. But these are very, very old, and they represent a fundamental kind of organization in the genome. And so their presence in the archaea as well as in the eukaryotes is really quite important. There are a number of different ways that the archaea are different from the bacteria. So for example, there are some details about transcription and translation that are different between the bacteria and the archaea, but these aren't differences that I expect you to know down here. If you look at the archaea, you find a lot of different things that are kind of unusual about their transcription and translation, but some of these are shared with the eukaryotes, again, signaling a very deep common ancestry between them. Finally, one other trait that bacteria and archaea share in common, a symplesiomorphy, is the fact that they both have circular chromosomes. In addition, you can sometimes find in these prokaryotes what are called plasmids, small supernumerary chromosomes or additional chromosomes, auxiliary chromosomes that are also circular in shape. Apart from these commonalities that I've been talking about, it's really hard to find a whole lot of commonalities among the bacteria. They are incredibly diverse. In size alone, you can compare the size of this circle here to the size of this one. The smallest bacteria are 8 orders of magnitude smaller than the largest ones. 8 orders of magnitude is the difference between 1 millimeter, which is about the thickness of the lead in a mechanical pencil, and 100 kilometers, which is the distance between here and Portsmouth, New Hampshire. This is a tremendous difference. In terms of shape, bacteria are incredibly varied. Their ability to move around also varies. Some of them can go through the water propelled by spinning flagella. Others thrash back and forth. Still others are simply content to float around. There's a tremendous diversity of bacteria, largely because this group is simply so old. You can see some of that diversity here, but we're really barely going to touch on it in this lecture. Unfortunately, we don't have time to cover all the amazing richness in the bacteria. We're just going to make a one-day dip into, we're dipping our toes for one day into the toe of this extremely diverse group. I just said that figure of speech completely wrong, but I'm just going to let it go. One type of diversity that you ought to be aware of in the bacteria, because it's quite useful, is what's called gram staining. This is a term that you'll run across fairly often in the literature. There are what are called gram-positive and gram-negative bacteria. Gram-positive bacteria have a lot of layers of peptidoglycan in their cell walls, whereas gram-negative bacteria have some peptidoglycan, but then they have this outer membrane as well on the outside of this peptidoglycan layer. The reason they're called gram-positive and gram-negative is that they stain differently when they're treated with something called a gram stain. And honestly, this is not a good taxonomic trait. Both gram-negative and gram-positive bacteria are paraphyletic groups. They're not monophyletic groups, they're not clades. But the reason why this is very helpful to know is that that additional layer of peptidoglycan that you find on gram-positive bacteria gives them considerable protection against certain antibiotics. So if you are going into medicine, the difference between gram-positive and gram-negative bacteria is that gram-negative bacteria can be killed by some antibiotics that will not touch gram-positive bacteria.